Hi, I'm Josh. Welcome back to another video on the channel. Today we're going to be going through eukaryotic cell structure, including all the organelles and what their functions are. So let's get into this. So the nucleus is the largest organelle in the whole cell. The nucleus contains coded genetic information in the form of a DNA molecule. Now this DNA directs the synthesis of proteins. DNA is made up of different genes, and these genes code for different proteins. Now, the DNA is contained in a double membrane called a nuclear envelope. This protects the DNA from damage in the cytoplasm. The nuclear membrane has nuclear pores. These nuclear pores allow molecules to move in and out of the nucleus. DNA is too large to move out of these pores, so it has to be transcribed into smaller mRNA, which is messenger RNA, and that is small enough to go through these pores in the nuclear membrane. So these DNA molecules that are too big, they are associated with proteins called histones, and they form a complex called a chromatin. This coils and condenses into lots of chromosomes, which you'll know from GCSE. Right, let's move on to the next organelle. The next organelle is the nucleolus. This is within the nucleus, and it's responsible for producing ribosomes. It is composed of proteins and RNA. RNA is used to produce ribosomal RNA, which is combined with proteins to form ribosomes necessary for protein synthesis. So that's pretty simple. Move on to the next bit. The next organelle we're going to be looking at is mitochondria. Now these are in all essential organelles. Mitochondria are the site of the final stages of cellular respiration, where the energy is stored in bonds is released, and it's made available for the cell to use, and it's in the form of ATP. The number of ATP in a cell is a reflection of the amount of energy the cell uses. Mitochondria have a double membrane. The inner membrane is highly folded into structures called cristae, and there's an interior fluid which is called the matrix. Mitochondria also contain their own DNA, meaning they can produce their own enzymes and reproduce themselves. Right, the next organelle we're looking at is vesicles. Vesicles are membranous sacs that have storage and transport roles. They consist of simply a single membrane and a fluid inside. Vesicles are used to transport materials inside the cell. The next organelle is a lysosome. Lysosomes are specialized forms of vesicles that contain hydrolytic enzymes. They are responsible for breaking down waste material in cells, including old organelles. They play an important role in the immune system, as they are responsible for breaking down pathogens ingested by phagocytic cells. They also play an important role in programmed cell death, or apoptosis. Right, the next part of the cell structure is the cytoskeleton. Now, the cytoskeleton is present throughout the cytoplasm of all eukaryotic cells. It's a network of fibers necessary for the shape and stability of the cell. Organelles are held in place by the cytoskeleton, and it controls cell movement and the movement of organelles within the cells. So the cytoskeleton is composed of three different organelles. Not organelles, components. These components are the microfilaments. Now these are contractile fibers formed from the protein actin. These are responsible for cell movement and also cell contraction during cytokinesis. This is the process in which the cytoplasm of a single eukaryotic cell is divided to form two daughter cells. The other component is microtubules. Globular tubulin proteins polymerize to form tubes that are used to form a scaffold-like structure that determines the shape of the cell. They also act as a track for the movement of organelles, including vesicles, around the cell. Spindle fibers have a role in the physical segregation of chromosomes in cell division and are composed of microtubules. Right, the final part of the cytoskeleton is the intermediate fibers. These fibers give mechanical strength to cells and help them to maintain their integrity. Next thing we're going to have a look at is the centrioles. These are composed of microtubules. There are two associated centrioles to form a centrosome, which is involved in the assembly of the spindle fibers during cell division. Another thing we're going to be looking at is the flagella. These enable motility. They can be used to detect chemical changes, and flagella are longer than cilia. Cilia can be stationary or mobile. Each cilium contains two central microtubules surrounded by 9 pairs of microtubules. The next organelle we're going to be taking a look at is the endoplasmic reticulum. The endoplasmic reticulum is a network of membranes enclosing flattened sacs called cisternae. It is connected to the outer membrane of the nucleus, and there are two types. So you've got your smooth and you've got your rough endoplasmic reticulum. So the smooth endoplasmic reticulum is responsible for lipid and carbohydrate synthesis, and it's also responsible for the storage of those molecules. The rough endoplasmic reticulum has ribosomes bound to the surface of it and is responsible for the synthesis and transport of proteins. The next organelle is ribosomes. Ribosomes can be free floating or attached to the rough endoplasmic reticulum. They are not surrounded by a membrane. They are constructed of RNA molecules and they are the site of protein synthesis. Mitochondria, chloroplasts and prokaryotic cells contain ribosomes because they all require lots of proteins. The next part of the structure of a eukaryotic cell is the Golgi apparatus. It is similar in structure to the smooth endoplasmic reticulum. It is a compact structure formed of cisternae and does not contain ribosomes. It modifies proteins and packages them into vesicles. Vesicles leave the cell and lysosomes stay in the cell. Alright, now I'm going to tell you how all of these organelles we've just described work together in order to form proteins. You'll look at this more in depth in the future, but this is just the basic way that all of these organelles function together. So right, proteins are synthesized on the ribosomes bound to the endoplasmic reticulum. They then pass into the cisternae and are packaged into transport vesicles. Vesicles containing the newly synthesized proteins move towards the Golgi apparatus via the transport function of the cytoskeleton. The vesicles fuse with the cis face of the Golgi apparatus and the proteins enter. The proteins are structurally modified before leaving the Golgi apparatus in vesicles from its transface. Secretory vesicles carry proteins that are to be released from the cell. The vesicles move towards and fuse with the cell surface membrane, releasing their contents by exocytosis. That's pretty simple. I like that. That really helped me to understand all of the function, because you can just remember that paragraph. You don't really have to learn each individual organelle step by one by one. You can just learn that massive paragraph and learn how they all work together. We're now going to have a quick look at plant cell ultrastructure. Now the first thing in here is the cell wall. It's made up of cellulose. It's freely permeable. It shapes the cell. The contents of the cell press against the cell wall, making it rigid. It has a defense role against invading pathogens. Now we're going to look at the vacuole within plants. Yeah, it's a membrane-lined sac in the cytoplasm containing cell sap. Large permanent vacuoles are used to maintain turgor. The membrane is called the tonoplast and it's selectively permeable. Plants also have chloroplasts. These are responsible for photosynthesis. They're found in the cells of the green parts of plants. They have a double membrane structure. The fluid contained within the chloroplast is called the stroma. They have an internal network of membranes which form flattened sacs called thylakoids. Several thylakoids stacked together are called a grana. Called a granum, and then in plural, if you've got multiple granum, you get a grana. And then the grana are joined by membranes called lamellae. Grana contain chlorophyll pigments. Photosynthesis produces starch grains. Chloroplasts also contain DNA and ribosomes, so they can make their own proteins and reproduce. Internal membranes provide a large surface area for photosynthesis within. If you did find this video helpful, be sure to smash that like button. Also, comment down below if you've got any questions, and subscribe to the channel if you are new and found this video helpful for your A levels. And there'll be loads more helpful videos in the future, so I'll see you in the next one.